Cattle fever ticks can carry a parasite that causes babesiosis, or cattle fever. The cattle fever tick was eradicated from the US in the 1940s. Since that time, a quarantine zone has been maintained along the Texas-Mexico border. Despite being eradicated from the US, cattle fever ticks still pose a significant threat to our cattle. Having not been exposed to the parasite, our cattle are expected to be particularly vulnerable to cattle fever.
Cattle fever disease is a highly lethal disease for many of the livestock species we are invested in here in the US. Some of the main things that we're trying to work on are developments of vaccines so that we can treat the cattle and prevent the ticks from being able to transmit cattle fever disease. We're also interested in other aspects of the tick's biology, such as its genetic distribution and its population biology. Can we identify where these ticks are coming from so that we know where to develop surveillance systems and other monitoring approaches that will enable us to defend from incursions of these ticks into the US?
I'm Deanna Bodine. I'm a biological sciences laboratory technician in Kerrville, Texas. I focus on doing molecular work for our genomics research unit. When we receive samples from collaborators, the first thing that we do to produce high-quality genomes is to extract DNA from the samples. Once we have high-quality DNA, we can then prepare libraries. What kind of libraries we prepare is going to depend on which sequencing instrument we're going to use. Once the libraries are ready, we add them to the sequencer. The instrument then does its job and sequences the DNA. Once that's completed, we extract or transfer the data off of the sequencer, and our bioinformaticians process the data bioinformatically.
By sequencing the genome, we're developing the molecular mechanisms and tools that are available to other scientists to develop vaccine technologies or targeted molecular genetics approaches so that we can treat the tick and make sure that it limits its ability to transmit the disease and also to enhance the host resistance to these ticks. The work that we do here is sort of acting like the foundational knowledge that we need to develop treatment mechanisms and other therapeutics to prevent the outbreak of cattle fever disease here within the US.